Hi everyone, this is lecture 7, transcription. It's found in chapter 5 of your iGenetics textbook. We're doing a transcription overview today. It's the beginning of the chapter 5, pages 81 through 86. So let's get started. Like usual, we have a quiz to start. The first question is, an RNA sequence is produced from a DNA sequence by the process of... Number 2, RNA polymerase makes an RNA copy of a DNA sequence by... Number 3, RNA polymerase would make what RNA sequence from the DNA sequence below? The top strand is the coding strand, the bottom strand is the template strand, and all of the DNA is from the middle of a transcribed region of the gene. And finally, RNA polymerase binds to what DNA region? Let's begin. This slide shows something called the central dogma. This idea was put forth by Francis Crick in the 1960s. He envisioned the flow of genetic information starting from DNA through RNA and onto protein. DNA would code the information, that coded message would be transcribed into RNA and then translated into the proteins that make up our cells. The word transcription sometimes confuses students, and it gets confused with the word translation. The word transcription in everyday kind of language simply refers to a copying down of information. For example, a court reporter might transcribe the court proceedings. Whereas the word translation in everyday language means changing languages. For example, English to Spanish, Spanish to English. It's changing languages. In a similar way, the central dogma idea is that the transcription of nucleic acids is simply copying the DNA message into the RNA message. Whereas translating, now we're taking a nucleic acid, the RNA, and translating it into a new language, the language of amino acids and proteins. Before we begin talking about transcription itself, I wanted to introduce the concept of a gene. We're in genetics after all, but this is a tricky concept because there is not a really well-defined definition for a gene. For our class, a gene is going to be a region of DNA that encodes for a functional RNA molecule. On this picture on the bottom, it's the part in green. The beginning is called the initiation, the ending is called the termination, and everything in between is the gene. This portion to the left-hand side of the gene is called the promoter. It's colored blue. The promoter you can think of as a region of DNA that acts as a genetic switch. It turns on or turns off the gene. Genes in prokaryotes and eukaryotes are basically the same. However, they are organized differently. In prokaryotes, often one long gene is shown here on top, but it encodes several polypeptides, or protein coding regions, which are shown here in dark blue. For example, this one long gene, this one long RNA, will produce many proteins. For example, protein alpha, protein beta, and protein gamma. Often these proteins have similar kinds of functions or work in the same biochemical pathway. This is called an operon. They all operate together. On the bottom is a eukaryote RNA. There is typically one gene that is expressed per RNA. On the left here is something called the 5' cap. On the right is a poly A tail. We'll talk much more about those later. Now, structure-wise, we talked about DNA quite a bit, so that should look familiar to you, the portion on the left-hand side of the screen. But if you compare it to RNA, you'll see that they are very similar. The DNA is double-stranded, RNA is single-stranded. However, RNA can fold up onto itself to form localized areas of double-stranded RNA, but it is produced as a single strand. The sugar is different, but not by much. The sugar in the DNA is a deoxyribose. Remember that 2' carbon does not have a hydroxyl group. However, on RNA, it's a ribose. The 2' carbon does bond to a hydroxyl group. The bases are similar, and yet there's one difference. The DNA bases are A, G, C, and T. In RNA, the T is substituted for U, or uracil. You can see they're both pyrimidines, T and U, but the uracil does not contain the methyl group like the T has. The process of making RNA is catalyzed by this enzyme called RNA polymerase. RNA polymerase is shown here as yellow, and you can see that it's bound to a promoter, which here is in green. This is the genetic portion, the genetic regulatory portion of the DNA. Once it binds to the promoter, it begins moving along the DNA template, again making only one copy, one strand. It copies the template strand, which is shown here on the bottom, and this portion up here is called the coding strand. You'll notice that the direction the RNA polymerase travels is the same as it is for DNA polymerase. It synthesizes the new strand in the 5' to 3' direction, and it moves along the template in the opposite direction, in the 3' to 5' direction. You'll notice that just like DNA helicase in replication, RNA polymerase can unwind the double helix. It then begins adding new bases and proceeds from our left to our right. The RNA forms a small region of hybridization with the DNA template, shown here, but as the RNA polymerase moves forward, the RNA is released from the DNA template and then exits out the back. This is shown in more detail here in the structure picture on the bottom. The blue is the DNA, the red is the RNA. You can see that that RNA is being processed out the back, sent out the back. When we zoom in and look at the reaction mechanism, you'll see that it's very similar to DNA polymerase. RNA polymerase also catalyzes a phosphodiester bond linkage. And the energy for this reaction, again, comes from the substrate, the nucleoside triphosphate. That triphosphate group is high energy. It links that phosphate to the previous 3' hydroxyl, and thereby forms the phosphodester bond, shown over here on the right. In the process, it releases two of the phosphates, that's called pyrophosphate, and it proceeds again in the 5' to 3' direction. Alright, well that is all for this lecture. I will see you in class, and study up on Chapter 5.